Hi, I'm Stephen Ness, and I'd like to talk to you about the CASP competition. We're currently deep in the CASP competition here. You can see that we have these targets here are just about due, and we have a couple more. You can see that we have T409 and T2410 that are due on the 18th. So, and that we have this soft deadline coming up as well. So exciting times. Here's a puzzle. This is a real drug-like molecule, and this is a real human protein. We know that two of them bind together. There's just one catch. The protein has 3 different pockets, or maybe even more, that the molecule could sit in, and they're scattered right across it. So which one is real? That single question is the prediction. Let's work it out. First, meet the protein, because DDB1 is genuinely one of the most remarkable machines in your cells. Think of it as a universal adapter for a molecular shredder. DDB1 sits at the heart of an enzyme that tags other proteins for destruction, and it's modular. Snap in one of dozens of interchangeable receptor parts, and the same machine now destroys a completely different target. One adapter, a whole family of shredders. And we can show that here. Oh. Okay, and then we have to just go here, reload this. Here's a puzzle. This is a real drug-like molecule, and this is a real human protein. We know the two of them bind together. There's just one catch. The protein has 3 or more different pockets the molecule could sit in, and they're scattered right across it. So which one is real? That single question is the prediction. Let's work it out. So first, meet the protein, because DDB1 is genuinely one of the most remarkable machines in your cells. Think of it as a universal adapter for a molecular shredder. DDB1 sits at the heart of an enzyme that tags other proteins for destruction, and it's modular. Snap in one of dozens of interchangeable receptor parts, and the same machine now destroys a completely different target. One adapter, a whole family of shredders. Here's the protein right here. This is viewing it in small. The same protein also moonlights as a DNA damage sensor, patrolling your genome for sunlight damage and flagging it for repair. And here's the twist that makes it a drug target. because its aim is reprogrammable, it gets hijacked from both sides. Cancer drugs like thalidomide work by gluing this machine onto disease proteins to destroy them for you, and viruses, hepatitis B, HIV, hijack the very same machine to destroy your antiviral defenses against you. Same shredder, opposite intentions. So where a small molecule lands on this protein isn't an academic question. It's exactly the handle that medicine and infection reach for. This is a whole family of ligases, and you can snap in one of dozens of interchangeable DCAF receptors, and DDB1 becomes a different CRL4, each destroying a different target protein. Structurally, DDB1 is built from 3 of these disc-shaped beta propellers here in blue, green, and orange with a little cap at the end, the magenta cap. 2 of them clamp together into a kind of open shell, and that cleft is where the receptor parts and the small molecules dock. 3 propellers, several surfaces, and that's the root of our problem. There's no single obvious pocket. The ligand, now the molecule itself. CASP simply calls it, well, it's a off-the-shelf compound from a chemical catalog, about 25 atoms, a flat benzyl group, a little oxidiazole ring, and a chlorinated fluorinated tail. Here's the irony. It has this whole imposing systematic name on stream, but 0 recorded biological activity. Nobody has ever reported it doing anything to any protein. There's no prior data lead on, no known binding site to copy, which is exactly what makes this a genuine blind test. You can see that over here, and we have a 3D visualization of it as well here. You can see the whole ligand. And I'm using Smol to display all of this. This is the crux on DDB1. 3 different pockets have been shown, well, even more, with real crystal structures to bind small molecules. And here we can see them here. These are the crystal structures. Well, this is different crystal structures of ligands superimposed on a common backbone. Watch where they are. Blue over here. There's the blue. Green over there. And orange over there. These sites are 30 to 55 angstroms apart, opposite ends of the protein. A pose in the wrong one isn't a near miss, it's a completely different answer. So for our brand new molecule with no prior data, which of these 3 is home? To answer that, we throw the whole modern toolbox at it. And this is where it gets interesting, because the methods don't agree. The machine learning scorers, the AI docking models, are confident they pick the orange site, the tightest known binder. The physics disagrees when we run full molecular dynamics simulations, actually letting the molecule wiggle in water for tens of nanoseconds and measuring the energy. It ranks that orange site last and favors the blue and green ones instead. And as a sanity check, the same physics rediscover the correct context of a known binder from scratch, so it's not just guessing. And the third vote, simply folding the molecule into the protein blind over and over, keeps landing on blue. 3 families of models, 3 different favorites. The only site that shows up on everyone's shortlist is blue. So what you actually submit with when your own methods disagree, you hedge but not blindly. You choose to trust the physics over the AI scores and lead with the green. C-terminal pose, but we place a model at every credible site, 5 models, 5 pockets, as insurance against being confidently, catastrophically wrong. CASP stores all 5 no matter what order they're in. And then we wait, because right now this is completely blind. The real crystal structure is sealed until after the deadline. When it opens, it's the referee. We score all 5 models against the truth, and finally we learn who was right, the AI scorers or the physics. On a real hard 3-pocket target. That's the whole game. Predict, then find out. Thanks for watching. And you can see here, this is the CASP, and we have the targets that are expiring over here, and it's exciting times. We're right in the middle of CASP. The next video will be about T2409, which is an Amadeus with U04, and then we'll have a video soon after that with T2410, when that, I'll be doing that tomorrow. Thanks for watching. Again, this is SNES, Dr. Stephen S. Woohoo. Talk about CASP. Thank you very much.